Hi guys, welcome to another video. It's Iris the biology tutor. Today we're going to talk about protein synthesis, the central dogma of biology. Coined by Francis Crick, the process describes how DNA is transcribed to RNA, which is then translated to amino acids, the building blocks of proteins. Proteins are vital for the health and function of you and me, responsible for nearly every cellular task, including maintaining shape and organization, receiving signals from external stimuli, and mobilizing intracellular responses. Proteins are the workhorse macromolecules of the cell. The following is a general analogy that can help students better conceptualize the process of protein synthesis. Let's think of the process like a working bakery that produces cakes. The bakery itself represents one singular cell. Notice that there are rooms in this bakery. The division of rooms represents the compartmentalization of the cell. Remember that in eukaryotes, certain functions are specific to their corresponding organelles, which are surrounded by intracellular membranes. This compartmentalization greatly improves the efficiency of cellular functions and prevents potentially dangerous molecules from roaming freely within the cell. Similarly, in the bakery, we can say that each room has walls and a door, like the membranes of each organelle, which keep certain functions separate and allow for maximum efficiency. On the highest floor of the bakery, the hardest room to access is the recipe room. This room represents the nuclear envelope, a highly regulated membrane barrier that separates the nucleus from the cytoplasm. We can say the safety cabinet is the nucleus, where the DNA is stored. However, before we can use the DNA, it's important to make a copy of it so that the original form is preserved and kept safely in the nucleus. The recipe book contains instructions on how to make all different kinds of cakes: chocolate, red velvet, blueberry, and many more. Just like how the central dogma can result in the synthesis of a variety of proteins. Thus, another reason why we would like to make a copy is to selectively choose the specific cake recipe we would like to make. In other words, it wouldn't make sense to make a copy of the entire hundreds of pages if we only need a one-page recipe, just like how it wouldn't make sense to make a copy of the entire genome if we are only interested in the genomic sequence of one protein. The process of making a copy of a recipe in the book is called transcription. The resulting copy is called mRNA, messenger RNA. RNA polymerase, represented by Chef R, is an enzyme that facilitates transcription. Chef R is the only worker that can enter the recipe room. After the mRNA is created in the nucleus, it exits from the pores of the nuclear envelope and reaches the cytoplasm. The cytoplasm is where the translation occurs. tRNA, or transfer RNAs, are molecular bridges that bind mRNA codons to the amino acids they encode. On one of the tRNA end is a sequence of 3 nucleotides called an anticodon, which binds to its corresponding mRNA codon. The other end of the tRNA carries the amino acids specified by the codons. Let's use Mrs. T to represent a tRNA. Mrs. T carries the ingredients which represent the amino acids. And the addition of all the ingredients to create the batter represents the addition of amino acids into a polypeptide chain. The workers that help with the baking move the batter into the oven, which is the final baking step. This is like the proteins in the cell that work with the ribosomes to build the polypeptide. When the finished polypeptide chain is released, the chain is separated from its tRNA and can drift out of the ribosome. Since shape determines function and proteins exist in 4 states, the primary structure may still need to undergo folding, processing, or combination with other polypeptides before it can do its job as a functional protein. We can represent these post-translational modifications with the role of packaging a cake. The protein can either be used in the cell for intracellular function or can be shipped outside to be of extracellular use. Mr. V here represents a vesicle that facilitates the movement of a protein through the phospholipid bilayer and out of the cell. Security workers outside of the bakery help restrict movement into and out of the bakery and allow Mr. V to exit the cell. The security workers here represent the cellular concept of selective membrane permeability. In a future video, I can talk about RNA splicing, a process in which specific regions of the RNA transcript are removed and flanking sequences are joined together. This splicing process changes the information content of the RNA transcript, which then alters the translation of that information into a protein. Comment down below if this topic sounds interesting to you. And that's all for today. Thanks for watching this video. Don't forget to like, comment, and share it with your friends.